Hello, welcome to my lab. This is the Langdon lab, where we study the effects of ocean acidification and global warming on corals. One of the focuses of my research is looking at the genetic variability between corals. So we go out onto the reefs or the nursery areas, and we get corals, pieces of corals that come from lots of different parents. Each parent has its own unique genetic makeup that makes it more or less sensitive to climate change and acidification, disease, and lots of other things. But in particular, we're trying to see if amongst this diversity of coral types on the reefs today, any of them are what I'm calling supercorals. Corals that already have adaptations that'll make them maybe a little or hopefully a lot more tolerant to the kind of warmer and more acidic conditions that they're going to be exposed to by the end of the century. We think this is not impossible because if you go out during a bleaching event like we had in 2014 and 15, you could see amongst a lot of white corals some colonies that had a nice golden brown color, which means that they had not bleached when all their brothers and sisters around them of the same species had bleached. So we know that there's some that have these better genotypes. 23 weeks ago, we increased the temperature of the water in the tanks to 30.5. We think that's slightly above the bleaching threshold for this species. And over the last 23 weeks, we started with 230 corals in this experiment, and now all but 20 of them have died. So the ones you're looking at here are the supercorals that we're looking for. So these are pale. They're what we would call partially bleached. There's this kind of a gradient. This one's nice and dark on the end here. And this one next to it is quite extensively bleached, but they're still alive. They're still what we call calcifying. They're still building skeleton, but at a reduced rate. But these corals have survived much more heat stress than they've ever experienced in the wild, even by the end of the century. So now the intention is to recover these corals. I'll now lower the temperature, and we'll see if they can get back to a nice healthy color. And then the idea of this experiment is I would share these corals with my colleagues that are involved in coral reef restoration. They would take them out to a nursery area, and within 6 months to a year, these little pieces here would grow to a nice bushel-sized colony. And then the hope is that as they grow, they'll sexually reproduce, so each one of these could produce thousands of babies, replenish the reefs with healthy corals, and corals that we've shown here have a much better chance of surviving future climate change.